Right now, an atom of carbon in your left hand might have once been inside the lung of a Tyrannosaurus rex.
Oh, easily. Yeah.
Like, it could have been exhaled by that dinosaur, absorbed by some ancient fern, buried for millions of years, and then, eventually ended up in the very food you ate for lunch today.
It is wild to think about.
It really is. And that isn't science fiction. I mean, that is the actual thermodynamic reality of how matter loops on Earth. But, for decades, we just didn't teach science that way at all.
No, definitely not.
Like, if you imagine stepping into a middle school classroom 20 years ago, you weren't thinking about the epic journey of a carbon atom. You were just handed a textbook and told to memorize the periodic table.
Right, it was this very static way of looking at a completely dynamic universe. The traditional curriculum, it was often described as being, well, a mile wide and an inch deep.
Yeah, totally.
Just prioritizing rote memorization of these disconnected facts. I mean, you
Yeah.
memorized the parts of a cell, you memorized a food web, you took a multiple choice test, and then
You promptly forgot it all by the weekend.
exactly. But when you step into a 7th grade life science classroom today, one that's using the Bioforge 1.2 curriculum, the feeling is completely different.
It really is. It doesn't feel like a factory for memorization anymore. It feels grounded, like you are actually stepping into a working scientific field station.
Yes.
And our overarching mission for this deep dive today is to decode exactly how this curriculum pulls that off. Basically, by focusing on a framework called the Crosscutting Concepts, or, the lenses of science.
Yeah. And to really grasp these lenses, we first need to look at the massive shift in how science is taught today. It's built on a framework called three-dimensional science learning.
Right, the 3D learning.
Yeah, and the Bioforge sources use a really fantastic analogy for this. Just picture a physical rope.
Okay, a rope.
Right. A strong rope isn't just one thick string.
Yeah.
It requires 3 intertwined strands woven together to actually hold any weight.
Okay, let's unpack this rope metaphor, because I think it perfectly sets the stage for you, the listener, as we get into this.
Yeah.
If we are building the scientific rope for a student, strand 1 is what we call science and engineering practices, or SAPs.
Right, the SAPs.
This is basically the action strand. This is what you actually do. So it's investigating, designing experiments, physically making models.
Arguing from evidence, yeah. Which is very different from just reading about an experiment that someone else did.
Exactly.
Then you weave in strand 2, which is the Disciplinary Core Ideas, or GCIs. So if strand 1 is what you do, strand 2 is what you study.
The actual content.
Exactly. It's the foundational knowledge. So the big principles about cell biology, ecosystem dynamics, genetics, that sort of thing.
Which brings us to strand 3, the entire focus of our deep dive today. Strand 3 is the cross-cutting concepts, the CCCs.
The CCCs, yeah.
If the first 2 strands are the action and the knowledge, these are the lenses you use to make connections across everything you learn.
And what's really fascinating here is that these lenses aren't just for 7th grade life science.
Right.
Whether you are a middle schooler looking at a microscopic cell or a professional astrophysicist studying the cosmos, you are using these exact same 7 cognitive tools to organize chaos and make sense of the universe.
Okay, I want to avoid just reading off a list of these 7 lenses, so let's actually put them to use right now. Let's imagine a real massive biological mystery that a 7th grader might encounter in the curriculum, say the Bering Sea ecosystem.
Oh, that's a great example.
Right. So right now, sea ice in the Bering Sea is fluctuating wildly, algae blooms are behaving strangely, walrus populations are totally out of whack. If we just look at all those facts at once, it's overwhelming.
Just noise.
Exactly. So we need to put on our first foundational lens, which is patterns. When you put on this lens, you are simply asking, well, what repeats? What trends show up?
And it's a great place to start because noticing patterns is fundamentally the first step in any data analysis. Before you can figure out why the walruses are struggling, you have to organize the chaotic data. And in Bioforge, students do this constantly. They don't just read a paragraph about evolution, they actually graph raw data on fossil distribution and inherited traits.
So they are looking at thousands of scattered bone records and actually mapping them out over millions of years.
Yeah, and when they do that, a visual pattern emerges. They literally see evolutionary transitions in the graphs themselves.
Wow.
They notice that certain physical traits, ones that support survival in specific environments, become steadily more common over time, while others just fade out. They find the rhythm in the noise.
Okay, so in your Bering Sea example, a student would map out the historical ice levels against the walrus populations to see if a visible trend appears.
Exactly.
And once you spot that trend, say, the ice goes down and the walrus numbers go down, human curiosity just naturally kicks in. You naturally transition to lens two, which is cause and effect.
Right.
You see the pattern and you ask, well, what made this happen? What is the underlying mechanism? But let me play devil's advocate for a second here.
Sure, go for it.
If I'm a seventh grader looking at a chart and I see that ice levels dropped in the exact same year that a specific species of fish migrated north, how do I know the ice caused the migration? Like maybe it was just a coincidence or a change in ocean currents or a new predator?
And that is the exact trap this lens is designed to catch.
Oh, really?
Yeah. Phenomena in nature often have overlapping, really complex causes. If a population of organisms suddenly booms or goes extinct, it is rarely just random bad luck. When students apply the cause and effect lens, they are trained to distinguish between mere correlation, so two things just happening at the same time, and an actual physical mechanism linking them.
Ah, so they have to prove the work. They have to ask, was this population decline driven by seasonal temperature ranges, or was it a decrease in specific food resources?
Precisely.
This lens forces the student to evaluate empirical evidence to find the true causal link, rather than just guessing based on a graph.
Which naturally leads us to lens 3, scale, proportion, and quantity. The driving question here is, how does size, time, or amount change what we notice?
Now, I have to admit, when I first read about scale, my mind immediately went to physical size. Like, I just pictured zooming in on something really small with a microscope.
Yeah, that's super common.
But the sources made it clear it's not just about getting bigger or smaller physically, is it?
No, it's much broader than that. Physical size is a part of it, certainly, but scale is equally about time and amount.
Oh, okay.
To truly understand how a system works,
Yeah.
you have to recognize that a biological behavior happening at a micro scale
Yeah.
dictates what happens at a massive macro scale.
Let's ground this in a specific curriculum example. The sources talk about students looking at stomata. For you listening, stomata are those tiny microscopic pores on the underside of a leaf that open and close to let gases in and out. So a student might use a microscope to zoom way, way in to calculate the density of those pores on a single leaf.
But the crucial step is that they don't just stay looking through the microscope.
Right.
They take that micro-level data, the amount of water a single pore loses, and use it to calculate the total water loss of an entire forest ecosystem.
That is wild. I mean, they are mathematically connecting a microscopic cellular structure to the macro-level humidity and water cycle of a massive geographic region.
They really are. They are zooming in to understand the mechanism and then zooming out to understand the global impact.
Wow.
If we tie that back to your Bering Sea mystery, a student might need to look at microscopic phytoplankton blooms, so the tiny scale, to understand the massive amount of carbon dioxide being pulled out of the Earth's atmosphere, the global scale.
That brings us perfectly to our next group, the systems lenses. Because once you decide on the scale you're looking at, a single leaf, a whole forest, or an ocean, you have to actually draw a boundary around what you are studying. And that is lens 4, systems and system models. You ask, what are the parts, the boundaries, the inputs, and the interactions? So, like, if I'm looking at the respiratory system, the systems lens forces me to stop looking at the lungs as just a pair of isolated balloons.
Right, they don't work alone.
I have to figure out how they are communicating with the rest of the body to even know when to inflate.
If we connect this to the bigger picture, a system is a group of related parts that make up a whole and carry out functions that the individual parts just cannot do alone.
Yeah.
Traditionally, you might memorize the 11 human body systems as flashcards. Like, here are the bones, here are the nerves, here's the digestive tract.
Right, and just totally separate chapters in a textbook?
But under Bioforge, you view the body as a hierarchy of interacting subsystems. You have to explain how these systems rely on each other.
Yeah.
I mean, you can't understand a reflex action by just studying a single muscle.
Right.
You have to trace how the nervous system detects a stimulus, fires a neural signal across a boundary, and interacts seamlessly with the muscular system to create a physical response. The model forces you to see the interconnectedness.
And once you define a system's boundaries, you have to track what is crossing those boundaries, which is lens 5, energy and matter. You ask, where does energy flow? And where does matter go?
Yes.
And here's where it gets really interesting, because when you look at the thermodynamic reality of biology, there is a fundamental split between how matter behaves and how energy behaves. Matter cycles, but energy flows.
It is arguably the most important principle in all of life science.
Yeah.
Oh, definitely. Think back to your opening example of the carbon atom from the T-Rex.
Yeah.
Matter, so the physical atoms of carbon, nitrogen, oxygen, water is never created or destroyed. It is just borrowed.
Right.
Atoms move repeatedly through biological and geological systems in continuous closed loops. Decomposers break down dead material and recycle those exact same atoms right back into the soil where a plant takes them up all over again.
But energy doesn't loop. Energy is a one-way street.
Exactly. Energy enters the Earth's systems from the outside, almost exclusively as electromagnetic radiation from the sun.
Okay.
Plants capture that sunlight during photosynthesis and lock it into chemical bonds. Then, when a primary consumer like a rabbit eats that plant, the energy moves up the food web. But at every single step, a huge portion of that energy is degraded.
The sources mention that this happens because the second law of thermodynamics dictates that energy transformations increase entropy. But wait, entropy is one of those scary physics words.
Oh, it really is.
For our 7th graders, and honestly for me, are we basically just saying that every time energy changes hands, a tax is paid to the universe?
I mean, that is a brilliant way to phrase it. Yes, a tax is paid in the form of low-grade metabolic heat.
Ah, okay.
When the rabbit runs, it generates body heat. That heat escapes into the air, radiates out into the cold of space, and can never be recaptured by the food web to do useful work ever again.
So matter is a roundabout, it's just constantly recycling. But energy is like a highway that ends at a cliff. You constantly need new sunlight entering the system because the energy is bleeding out as heat at every single step.
Yes.
This perfectly explains why there are thousands of plants at the bottom of a food web, but only a handful of apex predators at the top. There simply isn't enough energy left after the universe takes its tax.
And by applying that energy and matter lens, a student isn't just memorizing an arrow pointing from a grasshopper to a frog. They are tracking the hard physics of survival across an entire ecosystem.
Which naturally leads us to our final pair, the dynamic lenses. We've tracked what flows through a system. Now we need to ask how the physical parts of the system are actually built to handle those flows. That is lens 6, structure and function.
Right.
You ask, how does the shape or physical composition of something help it do its job?
A major focus for this in 7th grade is the cell membrane. And what is really wonderful about the curriculum is that it doesn't just hand the student a clean, stylized diagram of a cell membrane. No, it grounds it in history. It actually takes students back to the 1855 osmotic experiments by Carl Nageli and Carl Kramer.
I found this part of the research so fascinating because before this experiment, scientists basically thought cells were just like formless bags of jelly.
Pretty much, yeah.
They didn't know there was an invisible, highly structured barrier keeping the inside in and the outside out. And to prove it, these guys used decalcified chicken eggs.
Yes. They literally soaked raw chicken eggs in vinegar.
That's amazing.
The acetic acid in the vinegar reacts with the calcium carbonate of the hard outer shell, completely dissolving it. I mean, it releases carbon dioxide bubbles and smells quite strongly. But what you are left with is a giant squishy egg held together only by a delicate, semi-permeable membrane.
You essentially have a macroscopic cell the size of your palm.
Exactly. And students recreate this in the classroom.
Oh, they do?
Yes. They take these squishy egg cells and place them into different environments. So pure distilled water, moderate salt water, and highly concentrated corn syrup. Over time, they weigh the eggs. What they see is the actual physical mechanism of osmosis in action.
Okay, wow.
The egg in pure water swells up massively, while the egg in the syrup shrivels up.
Because water is physically moving across that invisible barrier to try and balance the concentration of solutes. This is like the ultimate example of structure and function.
It really is.
The membrane operates as a selectively permeable barrier. Its physical molecular structure perfectly dictates its biological function, which is regulating water balance and maintaining internal homeostasis. If its shape or structure fails, the cell bursts or dehydrates.
And this raises an important question, though, which brings us to the final cross-cutting concept, lens 7, stability and change. Natural systems are incredibly dynamic. They're not frozen in time.
Right.
So this lens asks, when a disruption happens, what stays balanced? What shifts? What transforms?
Think about what happened to you, the listener, right before you hit play on this deep dive. If you just ate breakfast, your blood sugar spiked. That is a disruption to your internal system.
Yes, exactly.
Your pancreas had to detect that change and deploy insulin to drag your blood sugar back down to a normal level. You're literally a walking balancing loop.
And feedback loops are the absolute heart of this lens. There are two main types you look for, balancing loops, like your breakfast example, and reinforcing loops.
Okay.
A balancing loop counters change and promotes stability. It pushes back against the disruption to return the system to a baseline. In ecology, a healthy predator-prey relationship often acts as a balancing loop to keep populations stable.
But a reinforcing loop does the exact opposite. It compounds the change, right? It pushes the system further and further in the same direction, leading to runaway exponential growth or exponential decline.
Yes, let's bring this all the way back to your Bering Sea mystery.
Yeah.
Students analyzing that ecosystem are using the stability and change lens to figure out what kind of loop is currently dominating the Arctic.
Ah,
As ice melts, it exposes dark ocean water. Dark water absorbs more sunlight heat than white ice. That extra heat melts even more ice, exposing even more dark water.
that is a terrifying reinforcing loop.
It is.
So the student is looking at the data to determine, will this ecosystem eventually find a new balancing loop and return to normal? Or is it locked in a reinforcing loop that will push it into a permanent climate transition?
And they are using the exact same cognitive tools that professional climatologists are using to answer that exact same question.
So what does this all mean for the actual student? We've covered these 7 massive lenses: patterns, cause and effect, scale, systems, energy and matter, structure and function, and stability and change.
Right.
Okay, let's test this. I am a 7th grader staring at a blank test page about a dying coral reef. I don't know the answer. I'm panicked. My mind is totally blank. How do I actually use this framework in real time?
Well, the Bioforge framework gives a very warm, grounded piece of advice for that exact moment of panic. It says, when you are stuck, do not try to think about everything at once. Pick one lens.
Oh, I like that. I like to think of it like sitting in the chair at the eye doctor. You know, when you look at the chart on the wall and it's a completely blurry mess?
Yeah.
You don't try to cram 7 different prescription lenses over your eyes at the exact same time.
No, that would be terrible.
The doctor flips down just one lens. They ask, is it clearer, better, or worse? 1 or 2. If it doesn't help, you flip it up and try the next one.
That is a perfect analogy. If you are staring at that coral reef question and you freeze, you don't need to know the final answer immediately. You just pull one tool out of the kit.
Okay.
You ask yourself, can I see a pattern in the coral bleaching data? No. Okay, let me try another lens. Can I track the flow of energy through this reef? Or can I identify a cause and effect mechanism for why the water is warming? You isolate the problem.
I really want to make sure you, the listener, walk away with that toolkit. Whether you are a student preparing for a test, a teacher guiding a classroom, or a parent helping with homework, keep these 7 statements in your mental back pocket. This is your troubleshooting checklist.
Yes, absolutely.
When you hit a wall, just ask yourself if you can do one of these things. Ready? Number 1, I notice a pattern. Number 2, I can explain a cause. Number 3, I can define the system. Number 4, I can zoom in or out. Number 5, I can track energy or matter. Number 6, I can connect structure to function. And number 7, I can describe what stayed stable or changed.
If a student can run through that checklist, they are no longer just regurgitating memorized facts from a flashcard.
Right.
They are actively engaging in the cognitive behaviors of a working scientist.
Yeah.
And I think that leaves us with a really provocative final thought to mull over.
I love a good final thought. Give us something to think about.
The true nature of science is that knowledge is never totally finished. It is always open to revision. The history of human discovery is full of moments where new empirical evidence emerged, and scientists had to fundamentally change their explanations of how the universe works.
Yeah.
I mean, we used to think cells were bags of jelly until we applied new lenses to the problem. The reason we teach these 7 cross-cutting concepts isn't just to help a student pass a 7th grade biology test. It's because these are the exact same tools that global researchers use to reevaluate, question, and rewrite our understanding of the world every single day.
The cross-cutting concepts are how Bioforge teaches you to see science like a scientist. Not as isolated facts, but as connected systems.